Thank you for the great introduction, Harrison, and thank you all for being here today. As Harrison mentioned, today we're presenting on our mass tags. Title of our presentation is approaches to increase the multiplexing of proteomics data acquisition, more specifically creating new tags to first run more samples in parallel, a process that we call multiplexing, and also to improve spectral quality, which is the quality of data used to identify and quantify peptides and proteins by mass spec. So we're going to start off today's presentation with a biological example. And the biological example that we provided is one of neurodegenerative disease, because at PTI, we're interested in applying our novel proteomics technologies to Alzheimer's disease. So in this example, we have a human brain. And whether this brain is diseased tissue or whether it's healthy tissue, the brain is composed of hundreds of billions of individual cells. And each of these cells contains 20,000 to 30,000 proteins, and may contain hundreds to millions of proteoforms. In biology, we'd like to be able to characterize entire tissue regions, such as brain, but also be able to zoom in and look at the individual single cells within these regions and understand their proteomes. You can imagine this is a pretty large scale of analysis that we'd like to be able to do. Current approaches are not able to handle this scale. If we were to take a cell or maybe a group of cells from that brain tissue, and we were to run it by an LC-MS system in a core facility, it would cost roughly $150 per run to do this. Now you can imagine if we were to unpick a human brain cell by cell, this would be pretty prohibitively expensive, not to mention extremely time consuming. If we introduce approaches where we can plex samples together, or we can take different samples, we can chemically label or barcode them, combine them, and run them together on the LC-MS, you can see how the cost per sample. So the time would decrease, allowing us to speed up his biological analysis. At PTI, we're really interested in technologies that will allow us to increase the throughput of proteomics with mass spectrometry. And when we think about this idea of increasing throughput, we often come back to the Skynet principle, which is that throughput is equal to the number of parallel peptides times the number of parallel samples. And this is not the only time today that you'll see this proteomics pyramid to introduce this concept of throughput. So we can think of existing proteomics technologies that tend to increase one term of the throughput equation and not the other. For example, really high in the parallel samples dimension is this example called 35-plex DEA. And in this example, we can take 35 samples, we can label them and run them all together in the mass spec at one time. So we're really able to generate a lot of parallel samples. However, we're only able to analyze one peptide at a time using this method, and that's because we're reliant upon DDA-MS, or data-dependent acquisition, which is a mass spec acquisition scheme that allows us to analyze peptides individually and serially. Moving across the pyramid, we can see an opposite approach that allows us to really parallelize peptides. And this approach is called label-free DIA. We're only able to run one sample at a time with this approach, but we are able to get a greater depth of proteome coverage because we're using a data acquisition strategy called DIA, or data-independent acquisition. And in this acquisition type, all the peptides or precursors within a particular M over Z range are analyzed at the same time using this method, getting us deeper proteome coverage. At PTI, we'd like to be able to create multiplexing technologies that allows us to parallelize samples and parallelize peptides, achieving these multiplicative gains in throughput. There are a few such examples, or a few examples that are already able to combine parallelizing samples and parallelizing peptides to some extent. And one example of this is 3-plex DIA. You might be familiar with this. This was the technology worked on by Jason Dirks and Professor Slavov's lab. And the 3-plex DIA were able to run 3 samples simultaneously while maintaining that depth of proteome coverage. Guided by 3-plex DIA and these other approaches, we hope to reach n-plex DIA. In this talk, we'll specifically talk about the mass tags that would create and to enable this. Now, before we talk about the mass tags we've specifically created for this purpose, I think it's helpful to talk about what are the current state-of-the-art mass tags that enable 3+DIA. Well, this tag is called MTRAC, and you can see here this is its chemical structure. An MTRAC is a chemical label that gets conjugated to different proteome samples, allowing us to run 3 samples in parallel. MTRAC comes in a set of 3, so you can see it comes as delta 0, delta 4, and delta 8. And these delta numbers refer to how many headed atoms are contained in each version of MTRAC. Now, in a practical application, each of these versions of MTRAC is labeled to different proteome samples, and then those samples are combined and run all at the same time in the mass spec. And we can tell which peptide belongs to which sample based on its position in M over Z space. Looking forward to the technology we're interested in creating at PTI, we'd like to go beyond the state-of-the-art to increase the plex size. So this will allow us to hopefully be able to multiplex more samples together, but also increase or maintain the quality of data. Now, I think it's important or helpful here to say or to define what we mean in this context by quality of data. In this context, we're referring to the quality of data as the quality of peptide fragmentation spectra. And this is a peptide fragmentation spectrum here, and Nikolai also showed one of those in his talk. And so if you're unfamiliar with peptide mass spec, what happens is a peptide is isolated in the mass spectrometer and it's fragmented, forming these resultant peptide fragment ions, and they populate a spectrum such as this. And the different identities of these peptide fragment ions that help us figure out what is the sequence or identity of the peptide present in the mass spec at a given time. So based off of the fragments present in the spectrum, we can infer that this is the sequence. And you may notice that there are different colored peaks here, or annotated peaks. The blue ions correspond to B ions, which are peptide fragments that contain the N-terminus of the peptides. And as you can see there's a little caret. So, for example, VA has a little caret, and that means that N-terminal peptide fragment is found in this particular graph. Now, there are also red ions, or red peaks here, and these represent Y ions, which are conversely peptide ions that contain, or peptide fragment ions that contain the C-terminus peptide. And learning D and Y ions will be helpful for later during Mark's portion of this talk. Now, there are certain elements of a peptide fragmentation spectrum, such as this, that can help us aid in identifying the peptide sequence. And we call these elements of spectral quality. One of these is the number of annotatable peptide fragment ions. The more E or Y ions present, the easier it will be for us to figure out what the sequence of the peptide is. Finally, what is the relative intensity of these annotatable peptide fragment ions? The more intense each of the peaks is and relative to each other, the easier it will be for us to figure out the peptide crescent. And lastly, what is the complementarity of annotatable peptide fragment ions? For every bond breakage in a particular at a particular spot in a peptide, is there either a B or Y ion present? And so having these elements can really help us assign what the peptide sequence is. Now, tying this back to our ideas of increasing throughput, we want to either preserve or increase spectral quality when we conjugate our new mass tags to peptides, or it will be essential to increase our throughput. If you recall here, we have our throughput pyramid, and if we reach the top of our goal is to reach the top of the throughput pyramid, parallelizing peptides and parallelizing samples. And here I've also pointed out where 3+DIA lies on the throughput pyramid. Now, if we increase the number of samples we want in parallel with our mass tag, but we see an accompanying decrease in data quality or spectral quality, we can see that we're actually not increasing throughput. Because while we might be increasing the number of parallel samples, we'd be decreasing the number of parallel peptides. So the throughput equation would not be satisfied. So if our new mass tags reduce spectral quality, we're not increasing throughput. We'd like to be able to maintain our data quality, maintain the number of parallel peptides, or increase our data quality, increasing the number of parallel peptides as we as we multiplex more and more samples. And this will help us reach our goal of multiplicative gains in throughput. So to reiterate, but also to orient us towards the research portion of this talk, today Mark and I will be talking about creating new mass tags to first multiplex more samples, and secondly, to improve spectral quality. Now, the first thing to do when we were setting out on this journey of testing out new mass tags was to explore what type of small molecule compounds could serve as a good mass tag. And so we worked with the Pentelute group at MIT, and we started off with these 2 model peptide sequences. Now, on these model peptide sequences, a number of mass tag candidates were generated. And you can see here that each of these tags contains 3 components. In green, we have a trifunctional building block. And then on top of that trifunctional building block, there were 2 spots where we could derivatize. So the red represents a carboxylic acid derivatization, and the blue represents a boronic acid derivatization. This allowed us to form a bunch of mass tags combinatorially, which was great because it allowed us to explore a sufficiently large chemical space with a relatively simple experimental design. The next thing to do from here was to test how do these candidate mass tags, how do they impact our ability to sequence these model peptides? So what is their impact on the spectral quality of the given model peptides? We ran these model peptides labeled with these new mass tags in the LCMS, and the results here are in this graph to your left. You could see on the each of the data points or points on the scatter represent a different mass tag. And on the y-axis, we have the Andromeda score of 1 peptide. And what this is essentially a measure of is how easy was this peptide to sequence when it was conjugated to that mass tag. In other words, what is the spectral quality of this model peptide when it's conjugated to each of the individual mass tags? The higher the spectral quality, the higher the score. And the same goes for the x-axis, but this would be that same spectral quality measure, but for the other model peptide. Now, we were really interested in seeing whether there were any mass tags that could increase the spectral quality of both model peptides. And you can see that there's one highlighted up in the corner in red. And so seeing its ability to improve the spectral quality of both model peptides, we wanted to look at it a little bit further. And so here you can see this is the sequence of that mass tag that is highlighted in red there. And it's called PSMtag at PTI. And here you can see its structure and where it would be conjugated to a peptide. The next thing to do was to understand how can we use this tag for multiplexing? Where are the different sites on this tag where we could install heavy molecules? And we did this in collaboration with Dave and team at Creagen. And if you'll recall, using heavy atoms was is the mechanism by which we could multiplex using MTRAP. So this was an important thing for us to do with PSMtag as well. And with Dave's and the Creagen's team's help, we were able to produce a 9-plex and a 5-plex set of reagents with this mass tag. The next thing to do after coming up with this doping scheme was to also think about what is the impact of PSMtag on a human proteome? Does PSMtag also increase the spectral quality of a proteome? Because ultimately, we are interested in applying PSMtag to the proteome. And so we wanted to make sure that its positive qualities from this model peptide screening could be extrapolated onto the proteome. So we apply PSMtag to the proteome using a standard NHS ester labeling, which is pretty well-known for well-known chemistry for labeling proteomes with small molecules and mass tags. And this typically necessitates using a relatively large molar excess of mass tag to peptide. And so what we found when we did this initial reaction was that a lot of the unconjugated tag would cope with select peptides within our peptide gradient due to the relatively larger size of this molecule relative to other mass tags that are available. Fortunately, we were able to alleviate this using ion mobility to effectively separate the excess tag from the peptides. And we did this on both our thermo instruments with FAMES and our timsTOF SCP with TIMS or trout ion mobility. And this figure is able to show us that we're able to analyze peptide ions or isolate our peptide ions separately from the tag byproducts, allowing us to analyze all of the peptides that we're interested in without analyzing our tag byproducts. So next I'm going to hand over the microphone to Mark, who is going to talk a little bit more about the impact of PSMtag on the spectral quality of the curricule.
Thank you, Maddy. So following those accusations that Maddy was just talking about, as well as many others, we've arrived at a point where for a single DDA run, label-free a DDA run using the PSMtag label, we could achieve roughly equal amounts of peptides identified. So I'm showing unique peptide sequences identified in either sample here. And if we focus in on the intersection here, these are peptides that were identified in both a label-free and a labeled condition. So if you take this intersection, we can begin to evaluate in a more systematic way whether the score performance that we saw with our two original model peptides that we used to select this compound actually translates to a larger set of peptides from the human proteome. So each of the points in this scatter plot is going to correspond to one unique peptide species that was seen in both label-free and tag. And for that, we can compute both its ratio of relative abundance on the x-axis, and that shows us that our two samples on the whole are relatively evenly matched by intensity. And we can look at the ratio of score on the y-axis, which shows a distinctive shift in favor of the tag peptides. And this equates to a median 50% score increase of the tag peptides versus the label-free. So this annotated spectral plot may look familiar if you've been paying attention. And to give a little bit more context to this was not entirely cherry-picked. It was selected from the center of that distribution I just showed. So in an attempt to illustrate what an average score improvement as a result of the addition of PSMtag looks like on a spectral level. So these two peptides, these two spectra, happen to share a complete wide ion series. You can see it in red. These are the C-terminal fragments, and they match up fairly well. The B-ion series, the N-terminal fragments, are shifted by the addition of the tag mass on the N-terminus. And we also see additional ion information in the B-ion series introduced as a result of the tag addition. So we see this B1 ion, which is fairly strong. And we also see the higher B ions, B5 and B6. And it's the addition of this new information from the B ion series that is sufficient to increase the score of this peptide spectral match to a 34 fortune. And if we are to aggregate all peptide identifications from our human proteome sample, then we can actually observe that the pattern of fragment ion generation that we just observed in that isolated example is actually reflective of a broader pattern of fragment ion production across all the peptides in our data set. So here I'm plotting the relative frequency of fragment production for all applicable ions in the B and Y ion series, respectively. And if we hone in on B ion series, we see several instances of things that we saw in our previous example. For example, that B1 ion, so that is very rarely observed in label-free spectra, but with the addition of PSMtag label, it becomes observable in about 90% of peptide spectra. In this gain, and the B ion series extends out into the higher ions as well, and so we attribute much of the enhanced score performance yielded by this tag to the stabilization of B ions. But we can also begin to look at fragment ion coverage on a per peptide basis. So there are a couple of ways we can look at this, and the first is just as a percentage of theoretical fragment ions that we're able to match with peptides. So here I've show you illustrated example of a case where we have identified 6 out of 7 Y ions and 2 out of 7 B ions. So this equates to a 57% fragment ion coverage overall. If we take this metric and apply it to our set of intersected peptides from before, we see that the addition of PSMtag leads to a distinct shift toward higher percentages of theoretical fragments matched in our peptides, with a significant proportion actually showing 100% theoretical fragment matching. So this is going to be, this metric is going to have one of the highest correspondences to score in a database, traditional database search context. But in other contexts, it can make sense to look at fragment ion coverage a different way, as coverage of fragmentation sites. So so fragmentation can occur between any pair of amino acids in the peptide. I've marked possible fragmentation sites with the lightning bolts, and each fragmentation site can be covered by a Y ion, a B ion, or both. But there are certain applications in which having incomplete fragment ion information is the main limiting factor. And these can include localization of post-translational modifications, and also applications like de novo sequencing, where we might be challenged to resolve the ordering of a pair of unknown amino acids in the middle of the peptide. So here I've shown an example where incomplete fragment ion coverage can still yield 100% fragmentation site coverage. And if we look at the proportion of peptides that get 100% fragmentation coverage in our proteome sample, we see that we have huge boosts to this metric as well by the addition of PSMtag. And I've broken it down by peptide length. We see that actually the greatest margin of improvement comes from peptides that are a bit longer, on the longer side. So these are traditionally very difficult to get complete fragmentation site coverage for, but with that extended B ion series that we just saw, we can have enhanced information from the N-terminal side that can flesh out the fragmentation sites in the middle. Another important characteristic of tags that we are quantifying is the retention time shifts that the tags can induce. So to set this up, in a standard tryptic digest, will yield peptides that end at either lysine or arginine. In a label-free context, these do not elute significantly different from one another. Their distribution of retention times is very evenly matched. But the reason that we're interested in looking at this difference in lysine and arginine terminating peptides is that corresponds to the number of tags that can be attached to them. So the tag will react with the N-terminus of the peptide, but it will also react with the lysine side chain. So lysine terminating peptides can bear 2 copies of the tag. And we know that having 1 copy of the tag induces a comparatively small and consistent retention time shift on the order of minutes, several minutes. But when the number of tags on peptide increase, then this retention time shift gets compounded, which has the potential to make data analysis by various means a little bit more difficult. Unfortunately, this is not an issue for DDA approaches. So these are going to be, DDA data is going to be search with more traditional database search software, which is traditionally, which conventionally does not leverage retention time or retention time prediction to a significant degree. So we can see this is a 2 nanogram injection of a label-free tag sample on the Orbitrap Astral. And we see that the relative proportions of lysine and arginine terminating peptides that we're able to see in this DDA sample are relatively evenly matched. But it's a different story when it comes to DIA. So DIA search methods conversely rely heavily on well-matched spectral libraries, and particularly very accurate retention time prediction for every peptide in the library. This is where we start to see reduced performance and challenges in identifying these lysine terminate peptides. So I'm showing now 200 picogram injections on both our timsTOF SCP and Oritrep Astral. We see that we have very good coverage of our gene terminating peptides on par with or better than label-free, but the lysine terminating peptides have a much lower identification rate. And we attribute this mostly to suboptimal spectral libraries. So one important component of spectral library generation, which I just mentioned, was retention time prediction. So we think that the complex retention time behavior with the lysine terminating peptides makes this hard to predict, and that's one of the components of explaining this differential performance. And also it can be, we've observed that the lysine terminating peptides also have some unique spectral characteristics that might bring them less in line with the standard libraries that we use. But the good news is that despite using these suboptimal libraries, our protein identifications scale very well with plex multiplexing. So taking samples from the previous slide, I'm now showing the overall number of precursors and protein IDs identified in both a non-multiplex sample versus on the left two panels, a PSMtag enabled 5-plex. And we see that we're able to scale from 1-plex to 5-plex with very little impact on the number of IDs attained per plex. So this is great, and this represents an increase in multiplexing capability from what was previously attainable with a tag like mTrick. And this is also this performance that we see from 5-plex also extends to 9-plex. So this currently using a 9-plex with the 2 Dalton spacing tags rather than 4 Dalton spacing requires more sophisticated spectral modeling to really enable. So currently we're only showing this for PTI's own software JMod, which Kevin will be talking about next, and going into the joint modeling approach that actually enables this narrower spacing and the resultant the 9-plex. But scaling looks very good for both a 5 and 9-plex. And currently, we hope it's just a matter of getting more accurate and complete spectral libraries to get our identifications to vary. So to summarize our work thus far, we took 576 chemical candidates that led to a mass tag that allows increased confidence of peptide sequencing. We have a 5-plex at 4 Dalton spacing and 9-plex at 2 Dalton spacing. And we have this detailed synthesis that we are publishing open source, and we're working with commercial entities for licensing and distribution. And we're also looking toward the future for the development of a Generation 2 tag. So if you recall, our original assay was predicated on 2 synthetic peptides, and we've expanded this to a pool that we now have of 30 custom-made synthetic peptides. And we're developing an assay that will allow us to take these 30 peptides and label them with 30 or more compounds per run. And working with our partners at Creagen, we'll have 10,000 or more tag compounds to test in the near future. But with this high throughput screening approach, this should be very doable within the coming year. Maybe we potentially test even more. So what we want to do with this new assay is take what we've learned from the first generation of tag development and enhance it further. So we want to select for qualities like spectral quality that we've been presenting on, see if we can maintain that and hopefully increase it further. It would also be ideal, for example, to identify tags that have a minimized or even negative retention time shift, and that could help them work better out of the box with existing DIA software. And there are many more properties that we can quantify and leverage for the development of tags for both specific specialized and general purpose applications. And at the end of this process, we'll have a ton of data for these ton spectra accumulated for these 30 model peptides. And we hope this will help us develop a predictive model for chemical structures that enhance these features. And Mike is spearheading this effort. He's the one who's telling me to stop talking. So you can talk to him, and he'll be very happy to give you more context on our future directions here. So I'm going to turn the mic back over to Maddy for our acknowledgement.
Lastly, we'd just like to finish with some acknowledgments. So particularly acknowledging the MassTags team at PTI, Sarah, Harrison, Nikolai, Keller, Mike, it was just pointed out, Jane, Corinna, Anisha, and Helenka. And then also to acknowledge our funding sources, and then also specifically shout out our collaborators at Creagen. I think Dave and Raj are here today. And then also the Pentelute lab at MIT who helped us a lot with the early phases of this project. And then also the rest of the members at PTI. But with that, thank you so much for listening. And we'll open the floor to some questions.
Thank you, Maddy and Mark. I'm Sarah. I'll be helping moderate questions today. I'll also be monitoring on our live stream on YouTube. So for anyone joining virtually, please do chime in with some questions. But I see we already have one here in the back, so I'm going to...
Hi, great talk, guys. The potential to screen like hundreds, well, tens of thousands of small molecules for tags in the future is really exciting. Do you have any plans, instead of kind of just the high throughput approach alone, do you have any plans to maybe train a model on the chemical structures to kind of leverage, train a model for your desired quantities, like the lack of retention time shift or fragment quality, to kind of, in addition to the high throughput method, to kind of have an intelligent design strategy?
Yeah, great question. That is precisely what we're trying to achieve. And we're pursuing both kind of top-down and bottom-up approaches to this, so we can test like larger mass tags that are kind of variations on our existing PSMtag, and we're also interested in testing smaller building blocks that might show positive spectral improvements that could be aggregated to form larger structures as well.
Yeah, I'll just echo a beautiful talk. So I guess maybe two related questions. The first one is, it's interesting that you lose a bunch of the label-free IDs, like 8,000, some dropped out, and you picked up 8,000 new ones, so on balance it looks even. I'm just curious if you have any insights about like what are the nature of those peptides that you're losing your IDs on? And then relatedly, when you have a missed lyse cleavage, presumably you get a second tag there, and is that part of them that are going to these missed cleavages? And then finally, I was just sort of curious, like you get this nice RT shift. And I was curious whether you guys are thinking about actually taking advantage of that. You're getting some chromatographic separation, and maybe that's a feature, not a bug. Just how you're thinking about that.
Yeah, great questions. So I think the first one, we did notice that there's a pretty big difference in the intersect. This is in the context of the identities of the peptides changing between label-free and tagged, right?
Yeah, I guess I'm just surprised, like there were only 4,000 PSMs that overlapped.
Yeah.
And you lost 8,000 relative to label-free, you picked up 8,000
So we did notice that something that it's definitely something that we still have to explore, figuring out whether there are particular qualities of the peptides themselves that kind of lead to them being, or within the different populations of peptides identified by label-free and tagged, what the difference, physical property differences between those peptides are. We've also been wondering also about what the potential biological implications of that are, but something to explore a little bit further for sure. I think I might have to ask for your help on the second question.
No, when you miss a lysine cleavage, if you miss cleavage there, you get an extra tag.
Yeah, so if we get, so if we miss a lysine cleavage, then we'll get another tag, which I'm not even sure if we've been able to identify peptides that have had 3 tags. We've done a little bit of preliminary work with Barnes-Seeman peptides, and what we'll find is that if we have more internal lysines, we'll just see like a relative drop off in the number of peptides we can identify, and I think that might also kind of be hand in hand with the chromatographic separation. In terms of the actual usage of the retention time shift for the lysine-containing peptides with 2 small molecules on them, that's a really interesting question. I feel like right now for us that has definitely felt like a little bit more of a hindrance in terms of our DIA retention time prediction, but we'll have to think a little bit more about how that could be utilized in a helpful or innovative way.
No questions online at the moment. Any?
Thank you. Just going back to that intersecting peptide Venn diagram, I'm one of the non-technical people that Nikolai is encouraging to ask questions. What's on the non-label free side? Is that just tag that hasn't attached, or what's the 8677 number?
8677.
On the Venn diagram.
Oh yeah, so this is the number of unique peptide sequences that were found to be labeled with PSMtag, but were not found to be present in the label-free peptide population.
Got it. And my second question was, did anything really surprise you or challenge you that you didn't anticipate in this set of studies?
Well, I think Mark and I will definitely have different answers to this question. Mark, do you want to go first? Okay. I would say some of the challenges that are not described, I mean, obviously we're going to present the results that are more positive results than like some of the setbacks that we've had. Something that I've been working on a lot is the approach to bulk cleanup with this, approach to bulk cleanup processes with this tag. And so I think that has been one of the challenging chapters of this work. I think going into it, I didn't have a lot of experience with bulk cleanup. And so, sorry, to give you a little bit more context, if you recall this slide we have a pretty large excess of this tag byproduct versus the amount of labeled peptides, this can cause some problems for our analysis. And so we've made some strides in particular methods that could help us mitigate this. One being ion mobility, another being changing our labeling such that we have closer to stoichiometric ratio of tag to peptides, stuff like that. But one of the other approaches that we want to be able to do is to be able to physically remove the tag from the peptides, or from the tagged peptides, in order to aid our analysis. And that has definitely been a really challenging chapter, just trying to exploit the different chemical properties of the tag relative to the tagged peptides, and figure out some creative ways to do that.
And I'll add from the data analysis side. So the tag is 308 Daltons, which is really pretty large. So it is unusual to be working with a mass addict of that large size, if we're considering 1 or even 2 copies of it. So as a lot of you have alluded to, that causes a lot of unpredictable or unpredicted maybe effects on the populations of peptides that we can identify with and without the tag. So the populations are shifted towards shorter peptides with the addition of the tag. And there's also the concept of the tag having much higher hydrophobicity, which has that whole retention time shift. So there have been a lot of things like that have been maybe not foreseen in the tag design process that we've been parsing out in the data analysis side.
We are.
Am I allowed to ask you a question? Oh, there's another question.
Yes, we do have one question here, and then Nikolai has something. And
I'd like to answer Holly's question because she asked it very well.
All right, I'll be very quick.
So I have actually a positive and a negative surprise. So the positive surprise is we screened relatively small chemical space, and finding a tag with such favorable properties for fragmentation was better than what I expected. So I think that was a very good surprise, and that also gives me a lot of courage for screening larger space and possible improvements. The negative side has been how much effort it has been to train a good spectral library. The good part about that is it is very overcomable. It is something that currently affects the depth of our results, but this is something that we can foresee fixing relatively soon and then getting much better results.
Thanks for the talk. It's quite fascinating to hear this much about proteomics coming more from the small molecule side, MS side. One question for amino acids that have the same mass, isoleucine, leucine, how do you distinguish them? And the other, if you have, I mean, initially you were alluding to this, that you also want to see modified peptides and really look into post-translational modifications. How do you identify amino acids that have been modified and that are not the usual masses that you would expect, or even some that are new and no one has seen them before?
Yeah, so a lot of that is addressed in the standard database search strategy that's really been around since like the 90s, right? So, for example, your leucine isoleucine question, that is resolved for the most part by relying on a protein database that runs through a simulated digest. So we are limiting our analysis in most cases to peptides that are known sequences within the human proteome. So that will, given the context of the amino acids around that, we'll localize it to one or a few proteins, and that will kind of resolve that issue there. And what was your second part?
About modified.
Oh yeah. Yeah, so in the database search context as well, basically we're limited to, in most approaches, to searching for modifications that we anticipate. For example, methionine oxidation, phosphorylation. And these can be applied, in database search, we would call them dynamic modifications, where we consider both the version of the peptide that's unmodified and the version of the peptide that's modified with all the given permutations of those modifications. And whichever has the best match is the candidate that we go with.
Okay, let's thank Maddy and Mark one more time.